Hi, my name is Colleen Hanlon. I'm a professor in the Department of Cancer Biology at Wake Forest School of Medicine in Winston-Salem, North Carolina. I wish I was there with you all today in England. Hopefully I'll get an opportunity to come back and visit you. But for now, we'll use some of the few good things that COVID gave us, I guess, and I'll tell you virtually a little bit about TMS and as an emerging tool for various substance use disorders. So I like to think of our lab. We do human clinical research using brain stimulation and brain imaging. I like to think of us not only as an addiction lab, but as a lab that studies the fundamental human push and pull between craving and control. And I show this picture here, and the title of the talk is Saving Adam from the Apple, as a reference to probably one of the oldest stories of Western civilization. And that is, of course, as you are familiar with, I'm sure, Adam and Eve are sitting there in the Garden of Eden, and Adam is told that he can have a life of long-term happiness as long as he doesn't do one thing. And that is, he can't eat the forbidden fruit from this tree. So Adam's going along, living a life of harmony, presumably. And then all of a sudden, a beautiful woman approaches him with this large, gleaming red apple and offers it to him. And this represents an irresistible cue. And so Adam has a choice. He can either go for the long-term rewards of extended happiness, or he can indulge in this immediate forbidden fruit that's in front of him. So we all kind of know the moral of that story. But what I think is important is it's not just a story that we're all told when we were young, but this balance between long-term thinking and the ability to resist cues in front of you for an outcome down the road is part of everyday human behavior. And so, in fact, perhaps at this conference, maybe last night between the first and second day of the conference, you went out with your colleagues and you had a good time together. And hopefully, maybe this doesn't happen to you anymore, but maybe when you were younger, you would go out with a lot of friends. And there always seemed to be somebody in the group that would say, "Oh, come on, one more round." And you might think to yourself, "Gosh, okay, I'll have another. It would taste so good. It would make me feel good. Maybe it'd be free. I'd be socially reinforced." But then hopefully maybe another part of you said, it's getting late. I have to drive home. My significant other is at home waiting for me. And so you have these two things. And presumably, sometimes it's okay to stay and have another. But if the balance of these two processes is off and the appetitive drive or the cue-reactivity wins, that is not good for sort of stable, well-balanced long-term life. But it's not just alcohol. We're surrounded by weird temptations that we have to look at our devices all the time, which I find them particularly odd because it's not always rewarding. And yet we're still drawn to look at devices, even though perhaps as in this picture, we should be focusing on driving down the road. And of course, food is the thing probably that most people are exposed to, perhaps on your lunch break or in the snack break. You're tempted by goodies and baked goods, and maybe you're just walking to work and you have a variety of donuts or biscuits available to you, and you have to make a decision. Should I have another or should I not? And these behavioral processes are governed by specific brain circuits that have been well established for a long time. We sometimes refer to these as frontal striatal circuits in the brain. The mesolimbic circuit is a circuit that is involved in reward and arousal. It's that go circuit that says, "Oh, okay, I'll have another." The mesocortical system is involved in executive control. Sometimes this is called cold cognition, and helps us sort of withhold our go signal. And then the nigrostriatal dopamine system is most famous for its role in movement disorders. And you may have heard some reference to this in some of the previous talks, that in fact, actually, there's a lot of overlap between the motor system and obsessive compulsive thoughts and other sort of habitual compulsive behaviors. For this talk, I'm mainly going to focus on the mesolimbic circuit and the mesocortical circuit as emerging targets for TMS and substance use disorders. And as I mentioned before, the scientific premise is that addiction is a neural circuit disorder, and it's a disorder that happens when the balance between appetitive drive and cognitive control is sort of out of whack, right? And so if we think of these two circuits as being important components to the process of living a well-balanced life, we could use a non-invasive brain stimulation therapeutic, or potentially something like deep brain stimulation, to either try to increase executive control or to dampen down cue-reactivity and that impulsive action. And our group uses a technique called transcranial magnetic stimulation in order to make that happen. So what is TMS? You may have gotten a primer a little earlier by some of the previous speakers, and you may know, of course, from your own practices and sort of scientific backgrounds. But what I just want to kind of give you are some few basic principles of how TMS works. So the general principle was by Michael Faraday, one of my favorite Brits, except for Dr. Voon, of course. And what Faraday showed in the mid-1800s was that if you have a current and you pass a current through a wire, take that wire and you wind it up, it will induce a magnetic field. And he actually did that. The picture in the lower right-hand corner of the screen is an image of his original coil, which is now in the British Museum in London. I have never seen it, but it's definitely on my bucket list. And what he showed is that if you pass current through this coil, this iron coil will become a magnet. And so in this way, electricity is creating a magnet. And if you take a magnet and you spin it, it will induce an electrical field. And we teach this in physics class as the right-hand rule. So if you have electricity that goes through a wire and you curl that wire up, you generate a magnetic field which is perpendicular to the windings of the wire. And likewise, if that magnetic field, let's say, is pointed up and you switch it on and off, this is what we're kind of doing when we pulse the TMS coil, you'll in fact induce an electrical field which is perpendicular to the pulsing. And so when you do this in TMS, the most basic thing we do is you take the coil. Again, TMS was actually also invented by a Brit, Tony Barker, in 1984. So it's actually still pretty young technique. And he showed that when you apply TMS to the primary motor cortex, you can get a motor evoked potential in the hand. And that motor evoked potential is proportional in size to the strength of magnetic field that you put into the coil. But one of the challenges with TMS, or maybe it's a value, is that it only goes about 2 centimeters deep, let's say. And that's sort of a rough rule of thumb, of course. Don't quote me on like major newspaper about that. It's all dependent, in fact, on the strength of the coil of the current going in. But a general rough rule of thumb is that for a normal dose, you can get about 2 centimeters deep with a standard, what they call figure-of-8 coil. Now, there's been a lot of innovations in TMS coil design over the last 5 to 10 years. And so some coils specialize in the depth and the breadth of the field itself, and even the shape of the field. We like to tell our participants, some of the more educated participants often ask about how much of the brain are you stimulating when you push this TMS coil on my head? And so we had an opportunity to be interviewed by National Geographic a few years ago. And the reporter asked the question very simply. She said, imagine the brain is like a globe. About how much of the brain's surface are you depolarizing with a single pulse of TMS? And that is an answerable problem. And so we did sort of the math, and we figured out that a single pulse of TMS at a standard dose that would evoke a contraction in your hand is depolarizing an area about the size of India. And so that's kind of a fun little fact for you. Another important TMS fact is that while really the cortical ribbon is where we're having our primary site of action, we know through basic neuroscience principles that depolarizing the cortex, if you get enough neurons to activate, you can also activate the deeper brain structures. And so this is a principle that can be measured. The mere fact that TMS to the motor cortex contracts the hand shows this principle, because as you all know, the corticospinal tract goes down and there's a synapse in the spinal cord. And so the idea that we get a contraction in the hand shows we can jump at least one synapse. And this is the kind of principle that we exploit when we think about where to put the TMS coil if we want to interact with both cortical and subcortical regions. The target for depression is the left dorsolateral prefrontal cortex, and that's part of this executive control circuit. And the target that's been, that preclinical scientists would say is probably the most fruitful for cue-reactivity disorders is the medial prefrontal cortex. A third principle is that's kind of being rewritten now is that the effects of TMS on the brain seem to be frequency dependent. So low frequency TMS, like 1 hertz, tends to sort of quiet a neural circuit, whereas high frequency, like 10 or 20 hertz, tends to increase cortical excitability or kind of excite a neural circuit. That's kind of what we thought at first when we were first doing this, and we as a group of colleagues brought this from basic science studies that had been done in rodents. But then we started getting a little more complicated using things like theta burst stimulation, which is a sort of patterned frequency. And we learned that, wow, it's actually really complicated. And it's not as simple as basic frequency dependent effects. So there'll be more on that, I think, in the next 5 to 10 years. But this kind of for a 25-minute talk is sort of all you need to know is that there's some idea we can push and pull the brain with different frequencies. And then the fourth principle that I think is really important, particularly for patients with psychiatric disorders, whether that's Parkinson's disease or alcohol use disorder, is that white matter and gray matter matter. TMS requires physicality, requires a population of neurons to compute the signal and push it downstream, and it requires myelinated axons to efficiently propagate that signal down the fibers into the next neuron. And so you might imagine that diseases that have a lot of white matter or gray matter degradation, the TMS, the amount of signal reaching subcortical structures, like we've shown here in some of these papers, is compromised. So TMS for addiction, I kind of started in this field maybe like 10 years ago, and there were barely anybody doing it. And now I'm happy to say that the world is like kind of getting really excited about it, so much so that in 2019, we were able to get together a group of 77 authors from around the world representing multiple continents, doing various types of TMS and TDCS as a tool for addiction medicine. So I urge you to kind of check out this paper if you have a chance. Feel free to email me and I can get it to you if you need a copy. And then the best news perhaps of 2020 is that a form of TMS was FDA approved as a treatment for smoking cessation. In this particular trial, they had 262 chronic smokers, 14 sites, it was double-blind, sham-controlled. Everyone received 15 sessions of active or sham TMS over the course of 3 weeks. They used a coil called the H4 coil. And as you can see here, the red sides show the area that has the biggest magnetic field. In this case, it's kind of the bilateral inferior prefrontal cortex. You might hear this coil occasionally called the insula coil, and that's because it likely can reach the anterior insula. But it's important to remember it actually also depolarizes the left and the right hemisphere. And that may well be why it's efficacious. They had a pretty good success rate in that individuals that received the real TMS, which represent the green bars in these graphs here, were significantly more likely to have quit smoking altogether for 4 weeks after the intervention. And they verified this both behaviorally and with urine measurements. So that's really pretty great. The kind of summary there is that the efficacy of this first TMS treatment in the addiction field, it's pretty good. It's better than sham, but it's not really better than meds that are available. And so, so that opens some questions as to the specific group of individuals that this might be best for. Maybe people that are being treated for this may also sort of quit other drugs, so there could be some comorbid benefits there. We just don't know yet. It's still very early. So if we come back to these two constructs, we could either increase executive control or decrease cue-reactivity. I just want to show you a little bit about what's done. I don't have time to review all the literature, so briefly I'm just going to focus on alcohol. So to date, there have been 12 studies that in alcohol that have targeted the dorsolateral prefrontal cortex. Their sample sizes range from about 11 to 45. Of these studies, 5 of them changed drinking behavior, and then 5 of them changed a drinking-based biomarker. In general, that sounds great, but in fact, the 5 that changed drinking behavior aren't necessarily the ones that change the biomarkers, and they didn't necessarily both look at both of them. So it's promising, but still the studies are relatively small. When we look at the other idea of decreasing cue-reactivity and impulsive action, there have been 2 recent studies that have been published just in the last 6 months, which is really exciting. Harrell and colleagues did a study of 51 individuals with alcohol use disorder, which is the biggest study to date. And then our group, McCallie and all, published a study with 50 users. And so I'll tell you a little bit about these 2. The Harrell study used a H7 coil, I'm sorry. And it's a big coil that has a pretty large field. This is an estimated electric field model for this coil. And as you can see, it really targets a lot of the medial prefrontal cortex and more dorsal areas. In our study, the one below, we targeted the frontal pole. So this is an area of the orbital prefrontal cortex. It's also on the medial wall, but in this case, it's much lower. The take-home summary from this, if you want to divert your attention to something else or you're falling asleep, is that both of these were pretty comparable. They both had 15 and 10 sessions of TMS, about 3,000 plus pulses per session. The pattern was a little different. The coils were different. But in both case strategies, the follow-up visits, the individuals were significantly more likely to be sober or drink less at the 3-month time period. So why the medial prefrontal cortex? Well, we had shown in 2018 that we had a group of 156 individuals that had come into the lab. They were either smokers, drinkers, or cocaine users. And we showed them pictures of their substance of choice versus neutral pictures. And what we found in that study is that when we aggregated data from all of the individuals, it was these areas in purple, so the medial prefrontal cortex, as well as the anterior insula, that were most likely to be activated by the drug cues versus a neutral cue. When we divided that data up by the specific drug use category, like cocaine is in red, alcohol is in blue, and nicotine is in green. In fact, the nicotine and alcohol were really very strongly isolated to the medial prefrontal cortex, while cocaine was tended to be more sort of bilateral anterior insula. And as you can see in the cuts on the right, all the way up pretty far dorsal for the cocaine users. When we look at the alcohol users together, these red areas here are areas that across a population of 67 individuals, these are individuals that were not in that previous cohort, so it's another cohort. We can kind of replicate this idea that it's up here in the medial prefrontal cortex and the brain on the upper left, that you have a lot of cue-reactivity. And you also see it down here in the caudate and the putamen, consistent with those circuits I showed you in the first few slides. When you look at the distribution of individual variability on this on this plot here, I have everybody's individual hotspot. So the place that individual was most sort of reactive to the alcohol cues versus the neutral cues. And you can see here, it's kind of like all along the medial wall, sort of spread out. It makes it a little hard to capture with a really focused TMS coil. And so the group, this is a European and Israeli group, they use a large coil. So that's one strategy, right, is to say, well, we're just going to have a big field. We have a wide distribution of points. We're going to use a big field. So they use this H7 coil, and individuals came in, 51 of them, treatment seeking for 15 sessions over the course of 3 weeks. And they followed them up. A key thing here is when they're delivering the TMS, each session takes about 15 minutes, I believe. But right before they deliver the TMS, we all agree that it's likely important for TMS to engage the brain circuit that's involved in cue-reactivity. And so in this case, they take the beverage of choice and they put it in front of the individual. They have the individual hold the beverage in their hand and smell it, and then hold it in their hand during the TMS treatment session. So this was done for 3 minutes of holding and smelling, but not consuming the alcohol beverage. And the reason we do this is we have, in this case, a wide electric field. And what we want is the brain to kind of show us the pattern of what needs to be activated. And so if we kind of apply a wide electric field and then we let the brain turn on what's involved with that kind of memory of cues, then those areas get presumably reconsolidated and affected over time. And what they found was that the active group, which are the dark circles in this case, had a significantly lower percentage of heavy drinking days after 3-month follow-up. And they also validated that with biomarkers, and they found that there was lower ETG as well in the 3-month follow-up. In our study, we targeted the frontal pole, which is a little more ventral. Very similar study design. We took 50 individuals that were coming in for an intensive outpatient treatment program, and we delivered 10 days of real or sham, in this case, theta burst stimulation to the frontal pole, and followed them for 3 months. We also did cue-reactivity. In this case, we just did visual cues that were shown to them, and we did a kind of verbally guided imaginal exposure, like you might imagine is done for PTSD. A staff member instructed the participant to keep thinking about alcohol and the negative aspects of how it makes you feel during the session. And in this case, we found the real TMSs that are the dark lines that are on the top in this graph, that individuals that receive real TMS were 3 times more likely to just remain enrolled in the intensive outpatient treatment program, which itself is kind of a win, especially for our clinical psychology friends and therapists that treat them. And they're also 3 times more likely to refrain from heavy drinking, which we were able to verify with urine measurements as well. So that's really exciting. We also found that the brain reactivity to alcohol cues in the group that received sham, as soon as they left the intensive outpatient treatment program, the brain reactivity to cues increased over time, over 3 months. And then, but the group that received real TMS, their brain reactivity to cues decreased throughout the 3-month time point. So that was also very exciting. So this is great news. What's next? Well, the next real step is that we need to do a multi-site pivotal trial. In order to do that, we need industry, because academics like me don't usually go to the FDA. And so we need industry to get interested and incentivize them. I can't do that myself, but what we can do as a community of scientists is think more about refining participant selection and optimizing the methods. So just in the last few minutes, I want to show you a little what we're thinking about there. There's several prominent sources of variance. We often call this the elephant in the room. And one of them is scalp to cortex distance. There's a weird thing in the TMS field. We dose the TMS based on the motor cortex, but then we often apply it to the prefrontal cortex. And that's not necessarily always a good assumption that those things are going to be proportional. And then, as I mentioned in an earlier slide, there's a really big distribution of points. So not everyone activates this area right here or this area right here. People are different. And so Dan McCallie was that picture on the last side. He's done a lot of this work in our lab. He's now doing a postdoc at Stanford with Claudia Padula. And one of the cool things he's shown is that in our previously published trial, individuals that had an overlap between the TMS-induced electric field, that's kind of just the physics, and their own endogenous brain reactivity to alcohol cues, that's based on the individual, when they overlapped, those people had a much greater clinical improvement. So that's kind of a cool thing to look at in the future. We also find when we aggregate data, not only from our own cohort, but large sort of phase 3 and phase 4 information from big TMS depression trials, that women appear to receive stronger TMS-induced electric fields than men. And in fact, they tend to have better outcomes from TMS than men. This could be kind of just other sort of psychological aspects, but there's actually a lot of biophysical reasons this could be true. There are some hormone-based reasons to believe this is true, as well as just basic sex differences in anatomy, which is kind of cool. And then I think the last thing, which is probably most important, and I hope that the whole world thinks about this a lot, and you guys too in your own practices, addiction, of course, is not a monolith, right? And there's stages, and there's parts of your life when substance use is driven more by the hedonic aspects or cues. And then there's a lot of aspects of drug use behavior, which honestly are just habits, right? Smokers will tell you that it's just a habit. Like, I just need it. It's what I do. I smoke in the car when I go to work. I smoke at home when I walk the dog. It's part of a ritual. Alcohol users, alcohol also very much a ritual. And so those behavioral processes are really different brain circuits. And so if we want to be smart, we should think about the stages of addiction that individuals are in when we choose a TMS target. And that's all I have. Thank you all so much for your attention. I really appreciate it, and I hope to meet you in person sometime soon. My information is down here on the lower right if you have any questions. Of course, it takes a village, and these are all the wonderful people that have contributed to my work. Thank you all so much. Appreciate it.